Welcome to the Deep Dive, the place where we take a single complex topic, stack up the research, and really extract the most essential nuggets of knowledge for you. Today, we are focusing on one truly extraordinary molecule. It acts like a universal harmonizer, kind of seamlessly syncing your gut, your brain, and your entire immune system. We're talking about vasoactive intestinal peptide, or just VIP.
Yeah, and it's a fascinating one. It's a 28 amino acid sequence that genuinely lives up to its name. Well, VIT really is a very important peptide. We've got some pretty dense source material to navigate today, covering its, elegant chemical structure, its stunning multi-system functions, and maybe most crucially, its emerging role as a potential therapeutic agent. We're talking about some of the trickiest chronic conditions like neuroinflammatory and autoimmune diseases.
Okay, let's definitely unpack this intricate molecule, but before we dive right into the mechanisms and the biology, we absolutely have to start with a critical regulatory note. Our sources, they discuss various research compounds, and it's just vital we acknowledge the specific context of that research right up front.
Absolutely essential, yeah. The research world is very, very clear about the line between scientific inquiry and actual application. One source explicitly flags that the peptides discussed, especially those linked to certain distributors or specific studies, they are strictly for research purposes only. This is a really critical safety and regulatory point. They're explicitly not for human consumption or any kind of self-administration.
Right, not for human use.
Exactly. So our conversation today, it's purely an analysis of the scientific data, the mechanisms involved, and the potential within that formal research framework. Nothing more.
Okay, good. With that very clear boundary established, let's jump into the origins of this VIP messenger. We should probably start with the fundamentals. Where did it actually come from, and what is it made of?
All right, so we need to go back a bit to the early 1970s. VIP was first isolated from the small intestine of a pig. That's where the intestinal part of its name comes from. Structurally, it's classified as a neuropeptide. That just means it's a short chain of amino acids, a protein basically, involved in how neurons talk to each other. It consists of exactly 28 amino acids.
28. Okay. And what family is it related to? Because, understanding its relatives often tells you a lot about its potential job profile in the body.
That's a really good point. It belongs to the secreting glucagon family of peptides. Now, this family, they all share a similar sequence and structure, which lets them interact with a specific type of receptor, the class BG protein couple receptors. And that connection immediately places VIP alongside some real heavy hitters in physiology.
Heavy hitters, like what?
Well, its cousins include PACAP, that's pituitary adenylate cyclase activating peptide, super important for neuroprotection. And maybe the most famous one right now is GLP-1, glucagon-like peptide 1. Everyone's hearing about that for its powerful metabolic and weight loss effects. So VIP shares functional and structural similarities with these, giving it this incredibly broad systemic toolkit.
Wow, okay. So if VIP is such a critical player, where's the actual blueprint for it stored in our bodies, in our genes?
Yeah, exactly. The VIP gene is located on chromosome 6, specifically at position 6q25 in humans. This gene, this doesn't just make VIP directly, it produces a larger precursor protein, what scientists call a prepropeptide. Then our cells have enzymes that act like molecular scissors, processing that large chain. They snip it to release the active 28 amino acid VIP. And interestingly, it also releases another coproduct called PHM, that's peptide histidine methionine, at the same time.
So they come out together.
Right. This processing ensures both peptides are released simultaneously, likely to carry out coordinated jobs.
What's truly remarkable looking at the sources is the distribution. It's not like it's just stuck in one place, like the pancreas or something. It seems to be literally everywhere the body needs coordination.
Absolutely everywhere. VIP is synthesized primarily by neurons. It is a neuropeptide after all, but also, interestingly, by specific endocrine cells scattered throughout the body. You find really high concentrations in the central nervous system, especially the hypothalamus and cortex, but also the whole spinal cord. And then peripherally, the entire GI tract. The heart, lungs, reproductive system, even the kidneys and thyroid. It's like a universal solvent for biological communication.
Which must give it this essential dual nature, right? It has to be sort of bilingual, speaking the language of nerves and the language of hormones.
That's perfectly put. That's the core of its translator role we mentioned. Within the brain, it acts like a classic neurotransmitter, signaling directly between neural circuits. Fast communication. But out in the periphery, the lungs, the gut, the immune system, It behaves more like a hormone-like modulator or maybe a co-transmitter. It influences local cell function, glandular secretion. It's constantly bridging that gap between the central nervous system and the peripheral organs, making sure they stay in dynamic balance.
Okay, a cross-system communicator that's also related to these powerful metabolic regulators. It's quite the molecule. So if VIP is the messenger, we need to know how it actually, knocks on the cell's door and delivers its instructions. Let's explore the mechanisms of action.
Right. How does it actually talk to the cells? Well, the delivery mechanism relies on that perfect fit, like a key in a lock, between the peptide and its docking stations, the receptors. VIP mainly interacts with two specific types of G protein coupled receptors. They're both classified as class B receptors, VPAC1 and VPAC2.
VPAC1 and VPAC2. Is there any specialization between those two? Do they do different things or are they found in different places?
Yes, absolutely. And this specificity is really crucial, especially when we start thinking about therapeutics later on. Let's take the pancreas, for example, which is so central to metabolism. VPAC1 is mainly found on the alpha cells. Those are the cells responsible for releasing glucagon, which raises blood sugar. VPAC2, however, is mostly on the beta cells, the ones that release insulin, which lowers blood sugar.
Oh, okay. So that differential distribution lets VIP subtly fine-tune the whole glucose regulation system depending on which receptor it hits more.
Precisely. It gives it that balancing capability. And it's also worth remembering that PACAP, its close cousin, is also a highly potent binder to both VPAC1 and VPAC2, in addition to having its own specific PAC1 receptor. So there's some overlap there too.
Okay. So once VIP docks onto VPAC1 or VPAC2, it triggers a chain reaction inside the cell. You mentioned earlier that the signal is usually a balancing signal. How does that molecular ignition switch actually work? What happens next?
So the process is pretty standard for this family of peptides, which means it's highly efficient and regulatory. Binding to either VPAC receptor typically causes it to couple or link up with something called a G's protein. Think of the G's protein as the molecular switch.
And what does turning on that G switch accomplish inside the cell?
Well, the activated G's protein immediately turns on an enzyme called adenylate cyclase. Adenylate cyclase then starts churning out a really important second messenger molecule called cyclic AMP, or just CAMM. And high levels of intracellular CAMP, that's like the cell's universal signal for regulate and relax.
Okay, that makes sense conceptually.
Yeah.
Regulate and relax. But what does the CAMP molecule do to actually achieve that state of regulation?
Good question. The CAMP activates another key player called protein kinase A, or PKA. PKA then sets off a whole cascade of events, often involving the phosphorylation that's adding a phosphate group and activation of a protein called CREB. CREB stands for CAMP response element binding protein. And activating CR leads to what we call CRE-mediated transcriptional cascades.
Right, transcriptional cascade. That sounds incredibly dense. Can we translate that into sort of everyday consequences for the cell? What does it mean?
Absolutely, let's simplify. CREB is essentially a master regulator for gene expression. It controls which genes get turned on or off. So when CRB gets activated by that VIP signal via CAMP and PKA, it moves into the cell nucleus and basically dictates a new set of instructions. For VIP, this typically involves turning off genes that promote stress, inflammation, or contraction, and turning on genes that promote things like cellular survival, self-repair, immune tolerance, like boosting the production of anti-inflammatory signals or protective growth factors.
So it's telling the cell to shift its programming towards a state of peaceful equilibrium, basically.
That's a perfect way to put it, a state of peaceful equilibrium.
But the sources also mention that sometimes, in specific instances, VIP can use a different pathway, something called the GQ-mediated pathway. What's the functional difference there?
Right. While the G's pathway leading to CHAMP is the primary route for VIP's regulation and relaxation effects, it can sometimes engage GQ signaling. This pathway uses a different second messenger system involving phospholipase C, and it's generally associated with different downstream effects, often involving the release of calcium inside the cell. The G's pathway is definitely dominant for VIP's main systemic balancing effects, but having the GQ option provides, let's say, cellular flexibility. It allows for more diverse, context-specific responses where maybe calcium signaling is needed. It adds another layer to its action.
Okay, so it's primarily GSC-AMP for relaxation and regulation, but it has this G-calcium option in its back pocket. Now let's talk about the immediate sort of macroscopic effects of that signal. What does the body actually feel right away when VIP is released and hits those receptors?
Yeah, the three most immediate effects are really profoundly felt physiologically. First, smooth muscle relaxation. We talked about how CAMP gets increased. Well, that inhibits an enzyme called myosin light chain kinase. This enzyme is like the molecular trigger, the clutch for muscle contraction. So by inhibiting it, VIP forces the muscle to let go, to relax. This is essential for opening up blood vessels, for example.
And that leads directly into the second big effect, right? Vasodilation.
Exactly. Vasodilation just means the opening or widening of blood vessels. That's what gives VIP its vasoactive name. And it is an extremely potent dilator. The sources say it's dozens of times more effective than even acetylcholine, which is another major player in vessel relaxation. The third immediate effect is increased glandular secretion, especially of water and electrolytes. We see this most dramatically in the gut, which needs fluid for digestion, but also in other glands.
Relaxation, vasodilation, secretion. Okay. The more we talk about this, the clearer it becomes that VIP isn't just, a peptide. It's the body's ultimate systemic regulator almost. Which brings us perfectly to the real deep dive section. VIP as the systemic harmonizer across, let's say, 4 major domains. Let's start with what feels maybe most urgent right now given current research trends, the immune system and inflammation control.
Yeah, this is a huge area for VIP. It really acts as the definitive immune peacekeeper. It's an incredibly powerful anti-inflammatory and immunoregulatory peptide. And this isn't just a passive role. What's fascinating is that VIP is actually produced by activated immune cells themselves, T lymphocytes, macrophages, dendritic cells, so it acts as an internal feedback loop. When the immune system gears up for a fight, VIP is released right there in the midst of it, presumably to monitor the response and make sure it doesn't spiral completely out of control.
Okay, so if the immune system is getting hyperactive, maybe in an autoimmune situation or a big infection, how does VIP practically silence the alarm bells? What does it do?
It uses a really clever sort of two-pronged strategy to clamp down on inflammation. Firstly, it actively suppresses the release of the major pro-inflammatory cytokines. These are the molecules that really drive the damaging aspects of inflammation, fever, tissue damage, pain. We're talking about the big ones, the foundational signals like tumor necrosis factor alpha or TNF alpha, interleukin 6, IL-6, and IL-12. VIP basically tells the immune cells, okay, stop producing so much of these.
Stop fanning the flames.
Yeah.
And what's the second prong of the attack?
While it's suppressing the pro-inflammatory side, it simultaneously boosts the production and release of anti-inflammatory mediators. The most notable one here is interleukin 10, or IL-10. IL-10 is widely known as the body's major calming signal in the immune system. It actively inhibits the function of those aggressive inflammatory cells and promotes tissue healing and resolution. So VIP effectively stops the attack signals while accelerating the stand down and repair signals.
Wow. That seems like the absolute ideal strategy for managing conditions like autoimmune diseases, where the core problem is the immune system mistakenly attacking the body's own tissues.
It really is the foundation for establishing or reestablishing immune tolerance. VIP dramatically shifts the whole landscape of T helper cell polarization. Now, T cells, they're like the generals directing the immune response. We have types like Th1 and Th17 cells, which are highly inflammatory and are known key drivers of autoimmune damage in diseases like rheumatoid arthritis or MSO. VIP actively pushes the T cell population away from developing into these aggressive Th1, Th17 types. Instead, it encourages the development of Th2 cells, which are generally less inflammatory, and most importantly, regulatory T cells, or TREGs.
TREGs. They're the ultimate peacekeepers in the immune system, aren't they? The internal police force.
That's exactly right. TREGs actively suppress the function of other effector T cells, preventing them from attacking self-tissues or overreacting to harmless antigens. So, by promoting TREG development and function, VIP fundamentally strengthens the body's overall immune tolerance. And this isn't just theory. When researchers look at animal models where VIP is missing or blocked, those animals show hyperinflammatory responses. They get much sicker from inflammatory challenges. Conversely, giving exogenous VIP, adding it back in, protects animals against things like lethal septic shock and dramatically mitigates the severity of experimental models of autoimmune diseases. We're talking rheumatoid arthritis, inflammatory bowel disease like colitis, and multiple sclerosis models.
So it really looks like a core regulatory deficiency in those conditions that maybe, just maybe, we could correct therapeutically.
Well,
Okay, fascinating. Let's move from the immune system now to the brain. VIP also seems to act as a conductor for time itself within the nervous system. How does it manage our internal rhythms?
Yeah, when we talk about the brain, VIP has a really high concentration in a specific area deep in the hypothalamus called the suprachiasmatic nucleus, or SCN. The SCN is basically the non-negotiable master clock of our entire body. It's what syncs us and all our physiological processes to the 24-hour light-dark cycle.
And what's VIP's specific role within that master clock mechanism?
Its key role seems to be synchronization and maintaining cohesion. The SCN is made up of thousands of individual neuronal oscillators, little clocks ticking away. The VIP neurons within the SCN are crucial for making sure all these individual neuronal clocks tick together in a steady, reliable, synchronized rhythm. Without this VIP-driven cohesion, the internal clock signal starts to fray and weaken.
Okay, so it keeps the orchestra playing in time. What happens if it's missing?
Well, knockout studies, where they genetically remove either VIP or its main receptor in the SCN, which is VTAC2, they show exactly that. The circadian oscillations of core clock genes become really disordered. And behaviorally, the animal loses its consistent rhythms of activity and rest. It's like the conductor disappeared. The musicians might still be playing, but the timing is all off.
That immediately makes you think about things like jet lag, shift work disorder, disruptions to that core timing. But beyond the master clock, how does VIP affect higher cognitive functions, things like learning and memory?
Right, this is where the mechanism gets really elegant and maybe a bit counterintuitive at first. VIP is also found in important brain regions like the cortex and the hippocampus, which are absolutely critical for higher processing, learning, and memory formation. But here's the interesting twist. It's primarily found in a specific type of inhibitory interneuron. Now, interneurons usually act as the brakes in neural circuits, slowing down signaling.
Wait, so VIP is located in the brakes. That sounds like it would hinder memory, not enhance it.
Exactly, it seems counterintuitive. But these specific VIP-containing inhibitory cells don't primarily inhibit the main computational neurons, the pyramidal neurons that do the heavy lifting of processing. Instead, they preferentially inhibit other types of inhibitory interneurons.
Ah, so it inhibits the inhibitor.
Precisely. It's like releasing the handbrake by specifically targeting the mechanism that keeps the brake applied. This results in a net disinhibitory effect on the principal pyramidal neurons. Basically, it transiently removes some of the local inhibition onto the main processing cells.
Okay, I get it. So by inhibiting the inhibitor, VIP effectively enhances the network's capacity to process information and change.
But that's exactly the idea. By transiently lifting the brakes on these key pyramidal neurons, VIP enhances network plasticity, the ability of the network to rewire itself. It facilitates long-term potentiation, or LTP, which is widely considered the fundamental molecular basis for memory formation. And generally, it seems to sharpen cognitive processes related to attention and learning. Conversely, disruptions in VIP signaling have been linked to cognitive impairments, particularly in aging.
Wow. And does it also act as sort of a shield for the brain? Neuroprotection?
It does, yes. Beyond its role in modulating function, VIP exhibits quite strong neurotrophic effects. That means it promotes the health, survival, and growth of neurons. Research shows it can help protect brain cells from damage caused by various insults, like low oxygen states, hypoxia, or exposure to certain toxins. So it appears to be both an enhancer of brain function and a protector of brain structure in the central nervous system.
Enhancer and protector.
Yeah.
Okay, let's move to its most, well, descriptive function based on its name, the cardiovascular effects. It's vasoactive for a very good reason, apparently.
Oh, absolutely. Its vasodilatory power is frankly astonishing. The source material consistently highlights this. VIP is documented to be dozens of times more potent at relaxing the smooth muscle and blood vessel walls than acetylcholine. And acetylcholine itself is a major physiological regulator of vessel tone. So yeah, VIP does not mess around when it comes to opening up blood vessels. It's incredibly powerful.
What are the implications of that sheer power on the heart and the circulatory system as a whole?
Well, it creates a bit of a careful balancing act, physiologically. Systemically, by relaxing vascular smooth muscle pretty much everywhere, it can significantly lower overall systemic blood pressure. However, it also seems to act directly on the heart muscle, or perhaps indirectly via sympathetic nerves synapsing on the heart. This action tends to enhance both the force of heart contraction, that's natropy, and the heart rate chronotropy. So it seems to be managing both the resistance in the pipes, the arteries, and the speed and force of the pump, the heart itself.
That sounds like potentially an ideal design for maximizing oxygen delivery, especially during stress. Lower resistance, but keep the pump strong.
It does seem geared towards that. Crucially, by dilating the coronary arteries, the vessels supplying the heart muscle itself, it ensures better blood flow and oxygen delivery to the heart. Particularly when the heart is under stress or working harder, it helps the heart sustain that high output, even if the overall systemic pressure might be dropping a bit due to the widespread vasodilation.
But the source material is very cautious about systemic administration, giving VIP as a drug because of this power. Why is that such a problem?
Well, this powerful broad effect is precisely the issue in a therapeutic setting. If you were to just infuse VIP systemically into the bloodstream, the resulting dramatic drop in systemic blood pressure, what we call transient hypertension, can trigger a strong reflex response from the body's baroreceptors. The heart rate shoots up, that's reflex tachycardia, as the body desperately tries to compensate and bring the pressure back up to normal. This really highlights the inherent challenge, maybe even danger, of using such a broad-acting, potent systemic messenger therapeutically without a very precise way to control its delivery or target its action.
Okay, that ties the cardiovascular effects directly back to the therapeutic challenges we'll discuss later. Very clear. Let's pivot now to where the story originally began for VIP, the gastrointestinal and metabolic functions. It was first found in the gut, and it acts as the original gut traffic manager.
It really does. In the gut's own nervous system, the enteric nervous system, sometimes called the second brain, VIP, serves as a primary inhibitory neurotransmitter. Specifically, it's often described as the main non-adrenergic, non-cholinergic, NANC inhibitory transmitter. It essentially directs a lot of the show down there. It relaxes the smooth muscle in the walls of the gut and also relaxes the sphincters, those muscular valves between different compartments. This allows food to move smoothly through the esophagus, stomach, and into the intestine. It also significantly increases intestinal blood flow, which is crucial for absorbing nutrients effectively.
And its secretory power, you mentioned that earlier. How significant is that in the gut?
Oh, it's immense in the gut. VIP potently stimulates the secretion of large volumes of water, electrolytes, like chloride and potassium, and particularly bicarbonate. This secretion comes mainly from the pancreas and the epithelial lining of the small intestine. The bicarbonate secretion is especially crucial because VIP also inhibits excessive gastric acid secretion from the stomach. So it helps neutralize any remaining acid entering the small intestine, protecting that delicate lining from damage. It's fine-tuning both the movement and the chemical environment for optimal digestion and absorption.
The sources make a pretty chilling point about what happens when VIP production goes completely haywire in the gut, though.
Yes, the pathology provides the most dramatic and frankly terrifying evidence of VIP's sheer secretory potency. There's a rare type of neuroendocrine tumor, usually originating in the pancreas, called a VIPoma. As the name suggests, these tumors overproduce and secrete massive amounts of VIP. This excessive VIP stimulation leads to a devastating clinical picture known as WDHA syndrome. That stands for watery diarrhea, hypokalemia, dangerously low potassium levels due to loss in the diarrhea, and achlorhydria, a lack of stomach acid because VIP inhibits it. The diarrhea can be incredibly severe, liters and liters per day, leading to life-threatening dehydration and electrolyte imbalances if not treated. It's a stark illustration of just how powerfully VIP can drive fluid secretion in the gut.
Wow, okay, so a powerful regulator, but too much is clearly dangerous. Beyond the sort of mechanical flow and secretion, you also mentioned earlier that VIP has an incretin-like action, meaning it gets involved in glucose stabilization, like its cousin GLP-1.
Yes, exactly. It plays a subtle but seemingly crucial role in glucose homeostasis, acting somewhat like an incretin hormone. Similar to GLP-1, VIP stimulates glucose-dependent insulin secretion from the pancreatic beta cells. This means it encourages insulin release mainly when blood glucose levels are high, like after a meal. And this action appears to happen primarily through the VPAC2 receptor, which, as we discussed, is predominant on those beta cells. It clearly links VIP signaling to insulin release under the right conditions.
Okay, so it helps lower high blood sugar.
Yeah.
What happens if blood sugar dips too low? Does it help then too?
It seems to, yes. It helps maintain the balance. If blood sugar levels drop too low, hypoglycemia, VIP appears to help prevent it from becoming dangerously low by promoting the release of glucagon from the pancreatic alpha cells. Remember, glucagon raises blood sugar, and this action seems to be mediated primarily through the VPAC1 receptor, which is more abundant on those alpha cells. So you see, VIP is acting as this two-sided stabilizer, nudging insulin up when sugar is high and nudging glucagon up when sugar is low, helping the body maintain that tight, healthy glucose range.
That's incredibly sophisticated.
It really is. And animal studies strongly support how critical this metabolic role is. Research shows that loss of VIP signaling, either by knocking out the peptide or its receptors, leads directly to features characteristic of metabolic syndrome. We're talking increased obesity, insulin resistance, poor glucose tolerance. It really underscores its importance in metabolic health.
Okay, VIP truly sounds like the linchpin holding together our entire physiological equilibrium, neural, immune, metabolic, cardiovascular, that involved everywhere. This brings us logically to Section 4, therapeutic potential, clinical reality, and of course the limitations. We clearly have a powerful biological target here, but the big question is, how do we actually harness it safely and effectively?
Well, the scientific data from preclinical models, animal studies mostly, is just overwhelming. Decades of research consistently show robust efficacy for VIP or VIP analogs in a huge range of disease models. We see things like significantly reduced joint inflammation and damage in models of arthritis. We see resolution of experimental colitis by calming the inflammatory cell infiltration in the gut wall. And we see significant neuroprotection and functional improvement in models of neurodegenerative diseases like multiple sclerosis and even Parkinson's disease.
And the underlying mechanism in most of these models seems to be the same, right? Calming that foundational inflammatory signaling cascade we talked about.
Exactly. It almost always comes back to its ability to modulate that critical NF-kappa B CRE signaling balance. By activating the CAMET-T CRE pathway, VIP effectively suppresses the activity of those aggressive, pro-inflammatory NF-kappa B transcription factors. It shifts the cellular response away from inflammation and damage towards tolerance, resolution, and survival. The potential to treat the root causes of many foundational chronic inflammatory and autoimmune diseases is just massive, theoretically.
So, has this immense potential actually transitioned successfully into human clinical trials yet? Have we seen it work in people?
It has, yes, although the journey has been complex. One notable form is an inhaled version of a VIP analog called Avictidil. This inhaled formulation was tested, for example, in patients with active pulmonary sarcoidosis. Sarcoidosis is a chronic inflammatory disease that often affects the lungs, and those trials did confirm its immunoregulatory effects in humans. Specifically, they observed decreased levels of TNF-alpha and increased numbers of regulatory T cells in lung samples from treated patients, exactly the mechanism we predicted from the basic science.
Okay, so proof of concept in a chronic lung condition. And more recently, it got quite a bit of attention in acute care settings, particularly during the COVID-19 pandemic, right?
Yes, that's right. An intravenous form of Avictidil was investigated, often under emergency use protocols, in critically ill patients who are suffering from severe acute respiratory distress syndrome, or ARDS. This ARDS was frequently related to the cytokine storm triggered by severe COVID-19 infection. Now, while these were generally smaller studies and the data is still being fully analyzed, many reported promising outcomes. VIP appeared to help mitigate that destructive cascade of cytokines, improve oxygenation levels, and protect overall lung function in these desperately ill patients. It really underscored its potential role as an emergency peacekeeper for runaway systemic inflammation, particularly in protecting the delicate alveolar cells in the lung.
Okay, so if the underlying science works, and we have promising early human data in both chronic and severe acute inflammatory conditions, why isn't VIP or Aviptidil a standard, widely used drug by now? This must bring us squarely to the core technical hurdles, the pharmacokinetic challenges you hinted at earlier.
Precisely. There are two major roadblocks that have historically limited VIP's therapeutic development. The first, and maybe the biggest, is simple biochemistry. VIP is a natural peptide hormone. Our body is extremely efficient at breaking down natural peptides very quickly using enzymes called peptidases. As a result, native VIP has an incredibly short plasma half-life. We're talking just a few minutes, maybe even less than 2 minutes. So if you just inject it systemically, it's effectively gone almost immediately. This makes achieving a sustained therapeutic effect practically impossible without resorting to continuous intravenous infusion, which just isn't feasible for chronic conditions.
Right, it disappears too fast. And then there's the other issue we touched on, the fact that it does so many different things, that broad action leading to side effects.
That's the second major challenge, because VIP naturally targets both VPAC1 and VPAC2 receptors. And these receptors are found throughout the entire body, cardiovascular system, gut, immune system, brain, everywhere. When you administer it systemically, you inevitably activate all its actions at once. That includes the powerful vasodilation mediated largely through VPAC1, leading to those unwanted side effects like transient flushing, headaches, and potentially problematic drops in blood pressure, the hypotension and reflex tachycardia we discussed. We want the targeted anti-inflammatory calm or the neuroprotection, but we need to avoid triggering that widespread systemic cardiovascular response.
Okay, stability and selectivity. Those are the twin challenges. So the entire research community is now laser focused on finding clever ways to bypass these two roadblocks, right? What are the current leading strategies being developed to stabilize the molecule to make it last longer?
Yes. The focus is intensely on developing stabilized formulations and analogs. One major approach involves creating enzyme-resistant analogs. This often uses chemical tricks like substituting some of the natural L-amino acids in the peptide sequence with their mirror image forms called D-amino acids. The peptidases in our blood and tissues, the enzymes responsible for breaking down the peptide, often can't recognize or efficiently cleave peptide bonds involving these D-amino acids. This can drastically extend the molecule's half-life in the bloodstream from minutes to potentially hours or even longer.
Okay, swapping out some building blocks to make it tougher. What about targeted delivery? Getting it just where it needs to go instead of flooding the whole system.
That's the other major avenue. Developing advanced targeted delivery systems. This includes strategies like encapsulating VIP or its stabilized analogs within tiny protective carriers. Think of things like liposomes, which are like tiny fatty bubbles, or biodegradable polymer carriers, often referred to as nanoparticles. These systems can serve multiple purposes. They can shield the peptide from enzymatic degradation, potentially allow for oral delivery, and maybe most importantly, they can be designed to accumulate or release their payload, specifically at the site of disease, say in inflamed joints, the gut wall during colitis, or even potentially across the blood-brain barrier. The goal is sustained release right where it's needed, minimizing that unwanted systemic exposure.
But perhaps the most elegant solution isn't just making the original VIP last longer or delivering it better, but actually shifting from stability to precision designing drugs that only hit the specific receptor subtype we want.
That is increasingly seen as the future, yes. Instead of trying to just stabilize the original broadly acting VIP, a major focus now is on developing highly selective receptor agonists. The goal is to design molecules that preferentially activate either VPAC1 or VPAC2, but not both. This would allow researchers to harness the specific beneficial effects mediated by one receptor type while avoiding the side effects associated with activating the other.
Could you give us an example of where that kind of precision targeting might be really beneficial?
Absolutely. We know, for instance, that many of the desired neuroprotective effects and also the beneficial metabolic effects like stimulating glucose-dependent insulin release seem to be mediated more dominantly by the VPAC2 receptor. Meanwhile, the problematic vasodilation and hypotension seem largely driven by VPAC1 activation. So if researchers can develop a compound that is highly selective for VPAC2 like LBT3627, an experimental VPAC2 selective agonist that's being explored in models of Parkinson's disease, they could potentially activate those desired therapeutic pathways, neuroprotection, metabolic balance, without causing the profound VPAC1-mediated cardiovascular side effects.
That's true surgical pharmacology at the molecular level.
Wow, that makes the selective agonist approach seem absolutely essential if we're thinking about treating chronic diseases, where long-term systemic injection of a broad-acting agent just isn't practical or safe. Exactly. Which leads naturally to the final area of major innovation mentioned in the sources, exploring new, often more localized routes of administration. Intranasal delivery, spraying the drug into the nose, is being very heavily explored right now. For lung conditions like sarcoidosis or maybe even asthma, it allows for direct local delivery to the airways, maximizing the effect there while minimizing how much gets into the general circulation. And perhaps even more excitingly, the nasal passage offers a more direct route for drugs to access the central nervous system, bypassing the blood-brain barrier to some extent. So intranasal delivery is also being seriously investigated for CNS applications, potentially treating cognitive dysfunction, maybe even neuroinflammatory or neurodegenerative disorders like Alzheimer's or Parkinson's, by delivering VIP analogs directly towards the brain.
It really sounds like the research community is designing bespoke solutions for this incredibly versatile but challenging molecule. The idea is to deliver the local peacekeeper exactly where the fight is happening, whether that's the lungs, the gut, or maybe even the brain, without causing unwanted chaos in, say, the cardiovascular system.
That is the perfect summary of the current state of play. Transforming this universal harmonizer, VIP, from a fascinating biological curiosity into a viable precision therapeutic tool is the singular focus of so much VIP research today.
Okay, as we wrap up this really comprehensive deep dive, let's just quickly synthesize what we've learned. VIP truly seems to embody systemic harmony. It's like the conductor keeping the body's incredibly diverse and complex networks. The neural, the immune, the endocrine, the cardiovascular, the gut, all in perfect functional sync. From regulating the precise timing of our master clock to delivering calming signals directly to our activated immune cells.
Yeah, it acts as the ultimate homeostatic regulator, doesn't it? When any part of the system starts to overreact, whether that's through runaway inflammation, excessive vascular resistance, metabolic dysregulation, or neuronal excitability, VIP appears to be one of the fundamental signals trying to gently pull the entire organism back towards a state of functional equilibrium, back to balance.
But its clinical reality, its use as a medicine, has been really limited by that frustratingly short half-life and its very broad, very powerful spectrum of action. Yet we've seen the incredible ingenuity now going into shaping the future of VIQ therapeutics. The enzyme-resistant analogs designed to last longer, the sophisticated encapsulated delivery systems for targeting, and crucially, that accelerating move towards highly selective VPAC1 or VPAC2 agonists.
The focus has definitely shifted. It's moved from simply acknowledging VIP's immense power to figuring out how to precisely master its deployment for therapeutic benefit.
Which raises a really important, maybe even provocative question for you, the listener, to consider as these sophisticated technologies continue to mature. Given that we know VIP is so integral to synchronizing our master clock in the SCN and also enhancing network plasticity and learning in the hippocampus and cortex, how will the development of these highly selective, perhaps locally delivered, maybe even intranasal VIP formulations change our whole approach to chronic illnesses?
That's the key question, isn't it? We've spent decades trying to treat complex conditions like inflammatory diseases and neurodegenerative disorders with drugs that often target just one single cytokine or one specific neurotransmitter pathway, often with limited success or significant side effects. But VIP, conceptually, seems to address something more fundamental, a potential breakdown in basic systemic harmony and regulation. Could it be that the targeted application of next-generation VIP analogs, maybe VPAC2 selective agonists delivered intranasally, might just unlock entirely new, more holistic strategies? Could this approach succeed in treating those complex circadian-linked neuroinflammatory and neurodegenerative disorders that have proven so incredibly resistant to our current, perhaps overly narrow therapeutic approaches? It's certainly something to think about.
An elegant molecule demanding an equally elegant delivery system and targeted approach. Fascinating stuff. Thank you for diving deep with us today.
It was my pleasure. Stay curious, everyone, and maybe keep seeking that systemic harmony in your own lives, too.